Hi, everyone. Thank you for sticking around. And we're only 30 slides away from coffee. Bear with me. I would like to thank once again the organizers for this incredible job in this conference. It's always nice to. Oh, is that unmuted actually? Yeah. They've done an incredible job organizing this, and thank you. Yeah. My name is Panos. I'm currently a postdoc at the Department of Anesthesiology at the University of Michigan. But today I will be talking a little bit about some of the work that I was doing at Penn in Philadelphia while I was doing my PhD. And specifically, that work has to do with humans for a change, but the relationship specifically between the structural and functional connectivity as assessed via non-invasive neuroimaging. There we go. So this talk is tripartite. First, I'll just start with a little bit of background that I think will be important to get out of the way before we start talking about some results. Then I'll go through some results for a recent study that we published, and I'll just finish with a slide of future directions just to talk about the future a little bit. So if we start with the background, I wanted to talk a little bit, and you guys might be familiar with this, but I just wanted to talk a little bit about this field called network neuroscience, right? And the French polymath Poincaré put it really nicely back in 1902 when he said that which science captures certain of the things themselves, but simply relationships between them, and beyond these relations, there's no knowable reality. And this is what network science in general as a field tries to capture. And a network scientist apply this on neuroscience to study our favorite dynamical system, well, some of our favorite dynamical systems, the brain. And essentially, the way that this goes is you identify independent entities, which in the case of the brain are brain regions. And then we call those nodes in network neuroscience lingo. And then we try to assess relationships between them, connectivity relationships between them. Of course, that can be defined using a myriad of ways, whether anatomically or via functional coactivation patterns. And once you have defined your independent entities, which in our case are brain regions, and how they're connected to each other, then you can come up with a connectivity matrix where essentially each row and each column correspond to the different brain regions, and each entry in that matrix gives you a number that tells you how strongly connected those brain regions are. Getting a little bit more down to brass tacks, one of the ways that network neuroscientists have been assessing connectivity across brain regions has been via structural connectivity. And this is exactly what it sounds like, is identifying anatomical connections between brain regions and seeing how strongly brain regions are connected to each other. And the way that we typically do that is by shoving someone into the MRI scanner and collecting a specific type of MRI called diffusion weighted imaging, which essentially what it does is it is really good at identifying movement of water molecules across the brain. Turns out we have a lot of water in our brains, and that's what it really capitalizes on. And after we do that, then we can apply some advanced cryptography software where essentially we can deduce at each voxel the directionality. And voxel is three-dimensional pixel, essential for those who might not be familiar with the term. But essentially at each voxel, at each three-dimensional pixel, we can determine the directionality of water molecules across different predefined directions. That's what the ellipses here show essentially. It's kind of like how probable it is that water is flowing towards that direction. And after we do that for all voxels, then we can combine all of that continuous information to reconstruct three-dimensionally white matter tracts across the brain. And here, the different colors essentially represent different directionalities of white matter tracts. So if you look at the red colors, those basically, for example, across the corpus callosum, those indicate white matter tracts that go from right to left and vice versa. If you look at the blue white matter tracts, such as, for example, the corona radiata or closer to the insula, that basically tells you white matter tracts that go from bottom to top. And then lastly, for example, you can have green white matter tracts that basically are the white matter tracts that go from the back of the brain to the front of the brain. And after we have three-dimensionally reconstructed those white matter tracts along which water diffuses, we can then define the structural connectivity matrix where every row and every column, once again, represents our brain regions, and each entry on that matrix represents how strongly anatomically connected those two brain regions are, as defined from the reconstructed matter tracks. On the other side of the spectrum, a more indirect way that people have been looking into to define connectivity at those brain regions is functional connectivity. And this is a lot more indirect. This doesn't look at something anatomical, but this rather looks at functional co-activation patterns across brain regions. So similarly, the way that we do that is we acquire, with some of the MRI scanner, we acquire an MRI called functional magnetic resonance imaging that comes in two flavors: resting state, where the person just doesn't actively do anything, they just are asked to stay there, and task-based, where, as you can probably imagine, you ask someone to perform a task while you're recording their brain activity. And once we do that, then for each brain region, we can extract we can deduce the neural activation time series. And then if, for example, we want then to decide how strongly functionally connected two brain regions are, then we can correlate the functional time series, and they're strongly correlated, then we can say that those brain regions fire in tandem. And once again, we can come up with a so-called functional connectivity matrix, where every row, every column is a brain region, and every entry essentially represents, in this case, for instance, a correlation coefficient that tells you how much in tandem those two brain regions fire. All right, so these are the two kind of flavors of connectivity that people have been looking into, right? But a lot of people, especially over the last 10 years, have been trying to bridge the two, right? And trying to see how can we use the structural connectivity matrix to predict the functional connectivity matrix. Or in other words, how does the structural connectivity matrix shape and constrain functional connectivity? Of course, this relationship is bidirectional, but yeah, this has been a very important question over the last decade specifically. And for that purpose, over the last 5 years, I would say, people have come up with this metric called structure-function coupling that essentially assesses the relationship between structural connectivity and functional connectivity. So the way that we do that is as I described earlier, you compute the structural connectivity of a human brain, then you construct the functional connectivity matrix of a human brain, and then in order to compute a brain region's structure-function coupling, is you identify the brain region on each one of those matrices, and thereby identifying their structural connectivity towards all other brain regions and their functional connectivity towards all other brain regions, and you correlate those two vectors. And you can then say that the correlation coefficient across those two vectors is the structure-function coupling. So if you have a brain region that has high structure-function coupling, this is a brain region whose functional connectivity mirrors, from a correlation standpoint, the underlying structural connectivity. All right, with that out of the way, I just wanted to mention some of the results that we recently published on this topic. And the first thing that I wanted to mention is that this structure-function coupling is a very heterogeneous marker, right? Different brain regions have different structure-function couplings, to the point where, for instance, you can have brain regions in the primary sensory and motor cortices, the green blobs that you see over there, that typically tend to have a high structure-function coupling. Their functional connectivity reflects their structural connectivity pretty well. In humans, pretty well is a correlation coefficient of 0.3, but that's another story. We can talk about that later. But then you have other brain regions, such as, for example, the ones in the higher order association cortices, like the prefrontal cortex and the limbic cortex, where essentially there's a very nonlinear relationship between brain region structural connectivity and functional connectivity. If you know the one, you cannot necessarily say much about the other. And indeed, a bunch of publications have really shown that different heterogeneous expression in structure-function coupling across different brain regions, depending on where they are placed in the unimodal-transmodal gradient. And besides that, from a behavioral standpoint, the other studies have also pointed the importance of structure-function coupling by saying that if you extend it beyond the regional standpoint to the individual standpoint, then differences in structure-function coupling across individuals have been doing a pretty good job at predicting how those people will behave differently in cognitive tasks. And lastly, of course, as you can imagine, structural connectivity, functional connectivity changes across neurodevelopment. So structure functional coupling also has been shown to really change. When we talk about health, we should definitely talk about disease as well. And from a clinical standpoint, structural functional coupling has been very important because it has been shown to change, to become aberrant across a vast range of neurological and psychiatric disorders. But more importantly, it has been shown to do a really good job at accurately tracking disease-specific pathology and duration.
So
Yeah.
I was wondering how much you could explain also this difference between like this condition and health in the like strictly, for instance, functional or structural network versus using this company.
That's an excellent point. We're actually about to publish a review that tackles exactly that. It has been shown for most disorders that structure function coupling is better than the sum of its parts. It has been shown that it does a better job at tracking symptomatology than either structural or functional connectivity alone. And another really cool thing that people have shown is that if you have, for example, a condition like multiple sclerosis, right? It was shown that the first year, even though structural and functional connectivity independently changed, structure function coupling did not change, and neither did cognitive performance. But then the second structural functional coupling changed, that's when cognitive performance also changed. So that's just one of the many examples, but no, that's an excellent point, and I completely agree in that whenever you talk about structural functional coupling, you have to also say individually what the structural and functional connectivity do. But yeah, no, that's an excellent point. All right. So in that empirical study, there were two main questions that we just wanted to address, right? The first question was, how does structure-function coupling vary across the cortical hierarchy? And as I mentioned earlier, that has been partially answered, but we wanted to go one step further. And the second, more important question is, why does structure-function coupling vary across the cortical hierarchy? What biological markers can we identify that seem to be driving this change? All right. Yeah, that. So the processing pipelines and the methodology that we followed in this paper to answer those two questions was quite intense and quite an overkill. But that was one of the points of the paper, right? We just wanted to show that whatever we show is consistent across a lot of different processing pipelines and across a lot of different methodologies, because here reproducibility was really the name of the game. So we used two cohorts. One was the Human Connectome project, which consists of 100 individuals, healthy and related. A lot of people have used that cohort. And the second cohort was a significantly smaller data set collected at Penn of 14 individuals. And the reason why that cohort was great is because they had really high resolution diffusion imaging. And we used 6 different processing pipelines to analyze these data sets. But today, just for the sake of brevity and sanity, I will stick to the first one, which was an atlas-based approach, where essentially we took the cortex and we parcellated it into 400 brain regions, well-established brain regions, to study connectivity that way. And then the second way, which was significantly more intense, was using a voxel-based approach, where essentially we identify each cortical voxel and we define that as its own independent brain region. So right now, you don't talk about 400 brain regions anymore, you talk about 60,000 to 85,000 brain regions, because each person has a different cortical voxels. So going back to the first question, how does structure function coupling vary across the cortical hierarchy? The first thing that we showed is, let's define first what cortical hierarchy is. So one definition that we use is some 7 very well-established functional networks defined by Thomas Yeo back in 2011, where essentially we took the 400 brain regions and we grouped them into those 7 networks, right? Visual network, somatomotor, different types of attention networks, limbic, frontoparietal, and default. And those essentially span the unimodal transmodal gradients. So what we showed, as you can see from the box plots, is that unimodal sensory regions, so regions corresponding to one sensory modality, like the motor and the visual cortex, they have higher structure function coupling than every other network. But as you go towards more and more transmodal brain regions, networks that incorporate a lot of sensory modalities to integrate information and operate across different timescales or slower timescales, the structure function coupling decreases, and you end up with a limbic network, which has the weakest structure function coupling. If we repeat this exact analysis using the voxel-based approach, so essentially here we're grouping, let's say, the 82,000 voxels for that specific individual into those 7 networks. It's kind of difficult to see that from the box plots because there's just so many outliers, but the results, if you look at the ANOVA tables, you'll see that the results are very, very similar. You have the motor cortex, which essentially has the highest structure function coupling. The visual, which you can't really tell here, but it is pretty high as well. And then the limbic has the lowest structure function coupling, and everything else is in the middle. So in that sense, it qualitatively agrees with the atlas-based results. Second way that we used to define cortical hierarchy to again probe at that question was using a more neurobiological definition of cortical hierarchy. So here, instead of grouping the brain regions into the 7 functional networks that I mentioned earlier, we grouped them into 5 different networks that the only thing that they vary in is in their cellular makeup. So here they vary in their granularization levels. Essentially, you start with granular layers that have very well-defined layers 2 and layers 4 that typically receive input from the thalamus. And then as you go along this hierarchy, you end up with 8 granular layers that consist of very long pyramidal cells that just span through all cortical layers, and there's not really a distinct layer 2 and layer 4. And the main take-home message from this is that granular layers, which typically encompass sensory brain regions, as we saw earlier, have the highest structure function coupling. But as you go towards more and more agranular cortices, which typically span higher order association regions, and the motor cortex actually, but yeah, the structure function coupling decreases. And then you have the polar cortex, which is a combination of the two that has the lowest amount of structure function coupling. So essentially the conclusion here is that structure function coupling decreases along the granular gradients. We see the same thing with the voxel-based analysis, which again, here because of the outliers, you can really tell with the box plots, but if you look at the ANOVA tables, it will give you the exact same qualitative conclusions as the atlas-based analysis, where we have the granular cortex on the bottom showing the highest structure function coupling, the strongest structure function coupling. And then once again, the agranular and the polar that have the lowest. The definitions of cortical hierarchy that I've been using so far are very coarse, right? 7 networks versus 5 networks. So we also wanted to use a more continuous definition of cortical hierarchy to look at the exact same question. And for that purpose, we turned to definition that was proposed in 2015 by a guy named Revilis at a really nice paper, where essentially he and his collaborators, they came up with a functional gradient where they assigned each brain region with a different number that corresponds to where they're placed in the unimodal to transmodal gradient. So you start with visual, sensory visual regions and motor regions that lie on the lowest end, on the negative numbers. And then you have on the other side of the spectrum, you have the limbic network and then the default mode network, those transmodal networks that have the highest numbers in that placement. So we saw how the structure function coupling change across the principal function gradient. And again, we saw the exact same message here across both atlas-based and voxel-based analysis. Voxel-based, what you see here is high density plots, by the way, those are obviously not scatter plots, because it's 82,000 data points. But you see the exact same message, the structure function coupling decreases along this unimodal to transmodal gradients. So moving on to the second and more interesting question, honestly, we wanted to see, all right, so far we have shown that there is this very large heterogeneous expression of structure function coupling across the cortical hierarchy, no matter how you define cortical hierarchy, across definitions that are both neurobiological in nature, but also functional in nature. So why is that the case? That was essentially the second question. But before I answer that, I wanted to briefly mention the 2 biological markers that we invoked to try to answer that question. And the first one is intracortical myelination. Exactly what it sounds like, the amount of myelin in the cortex. And the way that we non-intuitively assess that is using a well-established approach that was proposed a decade ago, where essentially we take the T1 weighted and T2 weighted MRIs, and those are anatomical MRIs. Those are MRIs that basically show you in really good contrast differences between white matter, gray matter, and CSF. And if you divide the two, you can essentially project the ratio onto the cortical surface, and this turns out as a really good proxy of the amount of the intracortical myelination across the cortex. And the second marker that we used was excitation inhibition ratio, where essentially that tells you the amount of relative excitation versus relative inhibition that goes on the cortex at different time points. And we assessed that using the Hurst exponent. That's based on recent work that showed that the Hurst exponent, which is a marker that assesses self-similarity across time series, we can talk about that later because that was quite complicated, but essentially we showed that decreased Hurst exponent based on the functional time series is a good proxy of increased EI ratio and thus increased relative excitation. So the results that we see here is that essentially across the atlas-based analysis, structural function coupling seems to correlate very strongly with both intracortical myelin and the Hurst exponent. And what that tells you is that brain regions that are heavily myelinated are the brain regions that tend to display functional connectivity that is mirrored by the underlying structural connectivity. So there's not much deviation of the functional patterns given the underlying structural patterns. And this makes sense, right? Because the heavier the insulation, the less amount of freedom and deviation that you can have. And the second result, once again, I think intuitively makes sense, in that the brain regions that had the largest Hurst exponents, which essentially corresponds to more relative inhibition, so the more inhibited brain regions are the ones that also displayed higher structure function coupling, which again, I think makes sense, right? Because the more inhibition you have, the less the functional signal can deviate from the underlying anatomy. We showed the exact same thing with the voxel-based results. As you can see here, just a lot more data points. And then the last thing that we did was essentially try to look at the relationship between intracortical myelination, excitation inhibition balance as estimated by the Hurst exponent, and structure function coupling across each one of the 5 neurobiological networks that I mentioned earlier, the granular ones. And for that, we used multiple regression models. So for example, if we take the agranular cortex, we performed a multiple regression model where the independent variables were the 2 biological markers of interest, myelination, the Hurst exponent, and the dependent variable was structure function coupling. And we did that for all 5 networks. And we just tried to see when you correct for both of those variables, which ones continues to correlate with structure function coupling. And just very schematically, the last result that I wanted to show and the conclusion from this was essentially that there seemed to be this really nice transition of power where essentially when you start with agranular cortices, which as I mentioned earlier are characterized by long pyramidal cells that span across different cortical layers, in those layers, myelin seems to play the most important role in predicting structure function coupling, whereas excitation inhibition balance doesn't do as much. But as you slowly transition towards more and more granular cortices, you notice that there is this shift, this gradual shift, where EI ratio, excitation inhibition balance, seems to be predicting a lot better how the structure function coupling behave. So the take-home message from this is that essentially, different networks rely upon different biological mechanisms to operate. All right. With that said, there were 3 main conclusions from the study. Well, there were a lot more in that paper, actually. There was a lot more results that went on it, but those were some of the results. And here, the first thing that we showed was that structure function coupling gradually decreases along the sensory association hierarchy. The second point was that brain regions whose functional connectivity essentially mirrored their underlying structural connectivity were brain regions that were characterized by increased myelination and more relative inhibition. And the last result was essentially that as you go from agranular to more granular cortical layers, there seems to be this change in predictive power, this transition in predictive power from intracortical myelination to EI ratio in predicting structure function coupling. And just the last slide is I just want to mention a few future directions into how can we incorporate these results to further study whole brain models. And the first thing is, first of all, I think we should start incorporating a lot more microstructural information and neuromodulatory information whenever we create such brain models. The second thing is that this is not the end of the story. Of course, there are a million other biological things that you can study, and some that I think are very important, and to also incorporate in said models is glial-neuronal interactions. So those are non-neuronal to neuronal interactions and vice versa. I think a lot of people have not been studying that for good reason. I'm not saying that it's an aspect of laziness. But the glial cells are very difficult to capture. And then, of course, internal and external brain states, which is another thing that we've seen changes the relationship between structure and function. And the last thing that I think was pointed a little bit by this talk is what is the spatial scale that we choose to study the relationship between structure and function, right? You can have finer core scale, finer brain, you can define brain regions in the very finer scale, you can define them in the very core scale. And of course, it depends on the hypothesis. We can talk more about this in the session later, but those are a lot of things to think about. And of course, the last thing that I wanted to do was thank the neuroscience graduate group back at the University of Pennsylvania, and of course the lab that I was in with my PI Dan Bassett, where we did all of this work, and then of course all the collaborators that helped with the study, and my current employer at the University of Michigan and the Department of Anesthesiology and the Center for Consciousness Science for all their support, and of course you guys. Hey. Yeah, that's it. Thank you.